Action. Yep. So I'd like to thank everybody for coming today and for hearing about my talk on the CHO Edge system, where we're trying to do advanced process development to tune the product quality. And so when I first started in the industry, a common approach was just to screen as many clones as possible, and then to throw as many process levers at that clone as possible to get to the process that we need. What if there was a better way to do that? What if we could start from the beginning and design these vectors in a more predictive way than we're currently doing that, so that we can understand how they'll perform from the initial design all the way to process development? And so in this talk, I'm going to be talking a little bit about the company in general, so the Asimov overview, and then I'm going to move more closely into the CHO Edge system and how we're utilizing that, and then how we're using that CHO Edge system to actually do some tailor-made process development. And then finally, I'm going to go through a case study where we're tuning post-translational modifications for this biosimilar that we're working on for a partner. And finally, I'm going to show how we're using all this in the future to develop data-driven models for process development as well as cell line development. And so, like I mentioned, I'll talk a little bit about the Asimov overview. What we're doing is we're developing a suite of cells, genetic parts, models, and software. And so what that does is it starts with our GMP host CHO-K1 GS knockout cell line. And then what we're doing is we're developing a comprehensive library of genetic parts so that we can understand how all these genetic parts perform within that vector sequence to predict how they'll actually work. And once we do that, we're developing data-driven models utilizing a lot of data that we do from the cell line development to the process development stage so that we can figure out how do these genetic parts actually fit into this whole ecosystem. And to generate all that terabytes of data, we're utilizing our computer-aided design software called Kernel. In most other industries, such as mechanical engineering, there's SolidWorks that allows you to design parts and predict how those parts will actually perform before we actually use them. In the biotech industry, there is some lack of that. So we're using this computer-aided design platform that we developed called Kernel. And so we're developing an access model where we can do it externally as well as internally with our internal services. And so I'm going to talk mostly about the CHO Edge platform, but we're optimizing and establishing for both lentiviral vectors as well as AAV vectors so that we can optimize those as well. And then also moving that towards the RNA Edge system, where we're actually trying to optimize the plasmid production, IVT, and payload expression control. And so now that I talked a little bit about Asimov as a general, I'm going to focus more closely into the CHO Edge system and how we're utilizing that. Like I mentioned, this starts with our host cell line. And so what we're doing here is we're using our CHO-K1 cell line that we got from the ECAC depository, and we cloned out of that and suspension adapted it with our chemically defined media and then knocked out the glutamine synthase. We use CRISPR-Cas9 for this, and we're able to do that because we de-risked it by licensing from all the major players in the patent portfolios, and then we banked them under ICH Q5 guidelines so that we could use it for clinical testing. And here on the right, you can see an example clone that we were able to get out of one of our platform projects, where we can see we have high viable cell densities, greater than 35 million cells, while maintaining high viability across the 14-day run for higher than 95% at harvest, as well as higher 95% throughout the whole run. And so the next part is we wanted to develop this comprehensive library of genetic parts, which includes promoters, 5' UTRs, as well as other UTRs, as well, and polyadenylation signals. You can see here we have different bar graphs, and each line on these bar graphs represents a different genetic part. What we did was we plotted on a log scale a combination of all of these genetic parts, and so it gives us this huge design space where we're able to mix and match all of these different genetic parts. This allows us to optimize the vector expression to be tailor-made to a specific monoclonal cell line, rather than doing a normal one-size-fits-all vector approach that most of the industry does. And so we're also coupling that with our mRNA hyperactive transposase, and this allows us to increase the copy number of our optimized vector payload so that we can increase the expression even further and get maximal productivity from our vector sequences. So kind of moving into this software platform, what we're using in the software is it allows the person to have a full parts library that we've characterized on the right and mix and match those into a vector sequence so that you can optimize the expression. And then you can also predict how these will perform and simulate it. For example, over here on the left is just a simulation of the GS, heavy chain, and light chain. So by doing that to tailor-made to each molecule, we can actually develop the software and the predictions that are required. And then just a little more details in this example of that software. Here's an example of 5 different in silico designs that we did. And so we can adjust the promoter strengths of these 3 different parts and then change the order as well to tailor make that expression so that it's specific to each molecule. And so kind of to go back to it for cell line development, what we're able to do is actually go from transfection to high titer RCB within 4 months. And so this allows us to go from our host, our Asimov GS knockout CHO host, and then within a month, we're able to develop the stable pool, clone and characterize those, bank them, and then at the 3rd month, we'll do an Amber 15 clone screen, and then finally, we'll do analytical characterization and release the RCP by month 4. And we're all able to do that at our cell line development site in 201 Brookline in Massachusetts. And we're able to do that with all of the industry-leading equipment that allows us to do single cell cloning, bioreactors, and analytical development. And so looking more specifically into this vector design, this was an example we did for a certain molecule. You can see on the left, we have 6 different vector variants that we did with 4 different molecules. An example is if we use one vector, and you can see in gray on the far left with the map, we would find that one of those designs, design 3, would give the best vector expression. However, if we use that same vector design across 2 other monoclonal antibodies, as well as a bispecific antibody that's a homodimer, so we'd expect similar expression, we would see that only one of those, that design would only work for one of those other molecules. And so what this allows us to do is to develop predictions and tailor-made vector designs for each of these molecules by picking one of these out of these 6 designs. And you can see on the right, this adjusts our absolute titer for our pools for those 4 molecules, where we're able to hit between 2 to 6 grams per liter for all 4 of these pools. And when we take that to the clone stage screen, we were able to find up to 6 to 10 gram per liter clones across all 5 of these molecules that we tested here. We have a 3-chain BSAP. We're able to hit 11 grams per liter for the top clone, while maintaining greater than 8 grams per liter for the top 12 clones. And then with the 2-chain BSAP hitting 8 grams per liter, maintaining above a 6 gram per liter for the top 12 clones. And then for the last 3 monoclonal antibodies, maintaining above 4 grams per liter for the top 12 clones, while hitting around 6 to 8 grams per liter for all 3 of these antibodies. So now that I talked a little bit about the CHO Edge system and the cell line development side, I'm going to show how we're utilizing that for the process development capabilities. And so at Asimov, we're integrating multiple bioreactor systems into one workflow. So we have the Amber 15 48-way system that we use for process screening as well as cell line development screening. And then we'll move that to the Amber 250 to do high throughput process development, where we have 24-way system. And then finally, we'll confirm that on the conventional 2 to 10 liter system. And so this allows us to go in a timeline from releasing the RCB at month 0, moving it to the Amber 15 within the 1st month, the Amber 250 in the 2nd month, and by the 3rd month, we're able to transfer that process after confirming it on the 2 to 10 liter. So the first step is we wanted to understand, is this scalable across the 3 different bioreactor systems that we're using, so we can demonstrate that our cell line and capabilities are scalable. So we have here on the left a MAB, and on the right the 2-chain BSAP, and we have the titer over time. We have in blue the Amber 15, in pink the Amber 250, as well as the purple, the 2-liter stirred tank reactor. We can see with both of these molecules, we had consistent expression over a 14-day fed batch bioreactor run, hitting around 6 grams per liter for the monoclonal antibody, and more than 6 grams per liter for the 2-chain bispecific antibody. We were able to maintain high viability across the 14-day fed batch run, while maintaining higher than 85% harvest viability across all 6 of these conditions. The next step is we wanted to transfer this process to our partners. And so we wanted to do 2 different things. The first thing we focused on was in pink, we wanted to see how does the partner process actually work at their facility compared to our process that's run at our facility in the middle, and then the partner facility on the right. And so you can see with the peak VCD and viability, we were at almost double the viability while maintaining higher peak VCD with our process regardless of where it was run. So we were able to demonstrate that we had higher viability in VCD with our process. And then when we take out the titer, we can see that we had a 54% increase from running the partner process versus both our facility as well as their facility for this molecule. For molecule 2, we wanted to actually take that a little bit differently because that partner wanted to go straight to the 12-liter. And so you can see here, we did the partner process again in pink. You can see that it had around the same VCD as our process. However, when we moved to the 12-liter at their facility, we had a slightly higher growth at 33 million, and that increase translated to a 20% higher titer compared to the partner facility using their process. So with both of these molecules, we were able to demonstrate consistent scalability and transfer of their process, outperforming their process. And so now that I talked a little bit about the CHO Edge process development, I'm going to move that to the case study that we did for tuning post-translational modifications for our biosimilar that we performed. And so talking a little bit about biosimilars, as you guys are familiar, they have very high titer demands because of their competition in the industry, as well as the need to match the originator product quality. And some of these product quality attributes are glycan profile, charge variance, and other critical quality attributes. And commonly, process development is used as one of these levers to actually modulate those critical quality attributes, as it's much easier to do than redesigning the cell line from scratch. And so with this example, we were doing the cell line development process where we had this 3-chain biosimilar that had 2 objectives. We need to maintain high heterodimer percent while also maximizing the titer. And so you can see here, this is an example of what we did where we generated 14 different vector designs from our kernel software. And so what we noticed is that if we had picked 1 or 2 of these 14 designs, the chances that we would hit either a high heterodimer percent and low titer or a low heterodimer percent and high titer would be very high probability. However, we noticed that we found 2 of these vector designs that were able to have both high pool titer as well as high heterodimer percent that we could move forward to the next stage. And so when we took it to the clone screen stage, you can see on the right that we were able to maintain high heterodimer percent from this pool for all 12 clones that we were able to find from this clone app. And you can see that it was above 70% across all 12 of the clones, as well as maintaining high titer of 8 grams per liter for all 12 of the clones, with the highest clone actually hitting 11 grams per liter with 84% heterodimer. So this demonstrated that not only was our vector optimization successful, it was able to be proven through the clone screen and go even higher than what we expected. And so kind of going into the details that I mentioned for the biosimilars, it's critical to match these critical quality attributes. So you can see on the right, these are the attributes that we typically test for. So we have found that we've noticed that aggregation, heterodimer intact mass, purity, as well as fragmentation were all within spec. However, we noticed that the charge variance and the glycan profile were slightly out of spec. So we wanted to focus on just the glycan profile for this talk and how we used our system to modulate that. And so we wanted to first start with our baseline. So what is our critical quality attributes for these top clones that we tested? So what we did was, you can see on the left, we have the top 7 clones that we tested, the relative titer between all 7 of them. And then we wanted to look at 3 different parameters that we noticed with the glycan profile. In the center, we have the normalized percent glycosylation, and you can see that all 7 of our clones were out of spec for that attribute. So we needed to adjust that. And this is relative to that green shaded area, which is the innovator molecule plus or minus 3 standard deviations. And so then we also noticed that we had a similar trend with the percent silation that was also out of spec. So that was something else we also needed to modulate, except for that top clone. And finally, as the normalized percent G0F is correlated to these other 2 glycans, you can see that this is similarly lower than what we were looking for all 7 of these clones. So this was something that we need to adjust or the partner would not move forward. And so what we're doing at Asimov is we're extending that vector optimization to also use our models to develop a quality tuning library. And how this works is we have an objective function, an example I'll talk about in a sec, and we have a bunch of different parameters, process parameters, median additives, as well as metabolic engineering that we're using with our modeling and analytics so that we can understand how to tailor a fit-for-need DOE and be a little bit smarter with the process development. And so for this case study, our goal was to reduce the silation and galactosylation while increasing the G0F. And so we have some other considerations that we usually take into account during this process, but I'm not going to go into it right now. So finally, our approach was to test two different additives. We wanted to look at that we identified through this quality tuning additive library. We found that N-acetylglucosamine, or glucnac, or glucosamine hydrochloride, glucan HCl, were two additives that we could explore to modulate the glycan profile. And so what we're able to show is on the left, the relative titer between, in orange, the control, in purple, the glucnac, and then in pink, the glucan HCl. And so you can see for the 4 clones that we tested, we had no difference in titer across these clones. So this demonstrated that what we were adding wasn't affecting the titer or any sort of expression machinery differently. And so we looked at adjusting the baseline, or understanding the baseline first again. So you can see in orange is the control. We had pretty similar results that we had with the previous run. So it showed that we had comparability between the two studies. And then we looked at how does the glucanac affect it. And we saw that we had a decrease in the galactosylation and silation. However, it wasn't within or close to those specs, so it was something that we wanted to explore further, but we did see that increase in the percent G0F that we expected. But then we looked at the glucose and HCl, we were able to decrease even further that galactosylation and silation. And that enabled us to get the normalized percent G0F within the specs for 3 out of the 4 molecules. So it showed that we're able to tune and tailor make this additive so that we can use it for adjusting that glycan profile. And so we wanted to make sure, does this scale from the Amber 15 to the 2 liters so that we can make sure that when we bring it to the larger scale, it translates effectively. And so again, we can see on the left, we have the relative titer, where we saw pretty much no difference between the Amber 15, which is in orange, versus the benchtop, which is in purple. So we saw almost no difference in titer, which is consistent to what we saw at that Amber 15 stage. And then when we looked at the percent galactosylation and silation change, we also saw pretty consistent results between the 2 scales, where it was for the first clone between 45 to 54% for the 2 scales for galactosylation and 71 to 61% for silation. And this correlated to very consistent results for that percent G0F change and increase between 37 to 40% between the 2 liters and the Amber 15. And all these results translated pretty closely to the same general trends for the clone 2, which demonstrated that we're able to scale this to the 2 liter. And so kind of putting it all together, within 3 months, we were able to develop a fine-tuned process that reached 11 grams per liter and able to match the innovative glycan species. We did this through detailed pool selection and cloneout, doing detailed critical quality attribute screening, optimizing those critical quality attributes for the glycans, and then confirming it and taking it out to our partner. And so now that I talked a little bit about that specific case study, I'm going to talk about how we're using all of that data to develop future directions and future predictions. And so in the typical process development world, we use design of experiments using partial least squares to generate different model correlations. So we'll design a DOE, calibrate a model so that we can get experimental titer versus predicted titer, and then use those insights to try to optimize or tune that design space so that we can tailor either the glycan species or any other species that we're looking for that DOE. And here at Asimov, we're trying to do that in a larger scale. We're trying to understand and develop bioreactor models so that we can increase our insights and develop even better predictions than just doing a DOE for each specific molecule. And so we applied a model-guided process development for 3 different molecules, and we were able to tailor-make that process in a much more efficient way that was able to increase both the VCD for all 3 molecules from 24 to 44% higher from the original platform, that increased the titer from 23 to 200% using this model-guided development. And how that works is that we're developing with our kernel system an end-to-end toolbox for model-guided CLD and PD. And you can see on the top left, we are using our genetic parts characterization system that I talked about with kernel, and then the genetic design system so that we can optimize those parts within the sequence. And then we're also coupling that with multi-omics modeling throughout the whole life cycle between cell line development and process development. And this allows us to optimize the bioprocess as well as the media design. And all this information we're coupling together to do some modeling and simulation of different aspects. We're modeling the gene expression, the phenotype so that we can make predictions, the metabolism within the cell, as well as understanding the bioreactor transport that's going on in our different systems. And all this allows us to optimize and tailor make our growth and titer and product quality so that we can be even more efficient with our process development. And so kind of just a summary of what we just talked about. We were able to deliver a high quality, tailor-made matching the innovator specifications. We did DNA design and synthesis using our kernel software, performed cell line development so that we can optimize and find that top clone. And then we did process optimization to meet our quality needs. And what it did was it achieved the target production of 11 grams per liter for this example within 3 months in process development. And finally, we showed that we're able to tech transfer and empower a robust process to those two partners that I demonstrated. And so this all generates and culminates to these 4 tenets that I talked about earlier that allows us to offer this disruptive CLD model where if we can't produce it at least 4 grams per liter, that whole program is free. And so I kind of just give acknowledgments to the whole team and everybody that's helped out with bringing this all together. And so this is everybody here at our rooftop at 201 Brookline. And then, yeah, I'd like to ask for any questions and thank everybody. If you have any more information, we have our website here. We're also going to be at booth 39 if you want to talk further. Thank you.
Thank you, Nicholas. That was a really interesting talk. And now we're just going to open the floor to questions. Yes, please. Can you use the microphone? Actually, I'll just give you this.
Nice presentation, thank you. My questions, I have a couple of questions. One of them is around the stability studies. Did you look at the stability of these cell lines when you tried a different plasmids that were predicted by the models? The other question is around the heterodimer percentage for the bispecific antibodies. When you tried the 7-day batch process, whatever the heterodimer quality that you are seeing, is that reflecting in the fed batch process when you tried in the fed batch process mode?
Stage.
Okay.
Yeah, so we have that data and we can maybe share it later if we need to.
Okay. Thank you.
Any more questions?
Yeah, and just to add, I did put in there that it was within specs. So that's kind of demonstrating that the heterodimer matched the quality that we cared about. We just didn't show it in this just to, focus on the adjustments that we need to do.
I'm interested in one aspect. So all this modeling and data prediction and sort of improving the yield and the product quality and characteristics, did that affect downstream process of purification? Did you see changes in, say, elution times and, say, fragmentation, different characteristics like that in the downstream parts?
Yeah, so for that project as well, we did do fragmentation analysis that the partner performed, and they didn't see any differences from their previous process as well as with any of the clones. So it was very consistent.
Excellent. Well, with that, I'll thank you again for your presentation.
Yep.
And I'd like to thank all of our this morning speakers. And for now, we're going to have a small little coffee break, and we'll resume our presentations at 11:10 in this room. And if anybody wants to continue discussion with any of the presenters, I'm sure they would be glad to answer any questions outside there.
Yep. Thank you very much for moderating.